So today we're going to talk about microbiological devices. We're kind of done with the more chemistry-oriented stuff that I have for you. So really for the rest of the course, we're just going to go through individual spaces of where you might think to apply synthetic biology and talk about some of the design constraints and opportunities in these spaces. So today we're going to talk about the area that deals with bacteria interacting with not really the environment, but specifically an animal sort of environment, a human body or a plant or whatever. So the scenario where you have cells interacting with other cells and all the complicated aspects of that. So these are things that control the interactions between organisms and their environment, be that soil, a cell, a plant, or an animal. And this week we're specifically focusing on the interactions with other organisms. And the reason why this particular subject is important is twofold. One, therapeutics are, at least with probiotics, a very real thing. And live vaccines, that's a very real thing. There are other opportunities in the area of direct therapies based on various cellular things, be that a bacterium, a virus, a mammalian cell. Lots of people work on this. We used to work on it too. So there are a lot of opportunities in the future. There's a potential growth area, put that way, for the field. So it's important for that reason. But the other big reason is biosafety. And if you become a practitioner of synthetic biology, you will get asked at some point in your career, probably multiple times, how do you know that what you're doing is safe? And I have watched students get asked that question and be like, I don't know. And it is an answerable question, kind of. It is answerable at the same level to which you can answer the question, how are you going to make that system? Where there's so much uncertainty due to the lack of characterization and encapsulation in this space. Like, we don't really know what things are going to do when you put them into the cell. So there, of course, is uncertainty about what exactly is going to happen. But there are several basic ideas that you should understand that are kind of the heart of what gives rise to pathogenicity and thereby danger. And as long as you are not creating solutions to these certain challenges that affect a microbe, then you really can't possibly be creating a pathogen. You could still be creating something dangerous, but not a pathogen. So we need to understand this. Okay. So in terms of how microbes are going to interact with other microbes or microbes interacting with animals, there are some basic biology ideas that I'm going to refresh for you, not because they are useful ideas, but just so this is how this area of describing biology is typically expressed traditionally. So like this old school biology. So mutualism is the scenario in which you have symbiosis, in which both species benefit. So it's resulting from prolonged interactions between the organisms. We're not talking about a scenario where you get diarrhea. We're talking about a scenario where a microbe stays associated with your body for an extended period of time, weeks and months, years in many cases. Commensalism is the scenario where one species benefits and the other is largely unaffected. Parasitism is the scenario where one species is going to benefit at cost to another. Okay? So you probably heard these things. An example of mutualism is like Vibrio fischeri and the bobtail squid, where you have a population of bacteria in a specific organ that, when squeezed in the right way, that population will glow. And that helps, actually I'm not quite sure exactly what the squid does with that, but it's somehow useful for the squid, and it's useful for the bacteria because they have a place to live and get food. Algae and lichens, these things can last thousands of years in symbiosis, like stable cellular interactions where you have an algae inside a fungus. Bifidobacterium in the human intestinal tract, the time that a microbe typically stays in your gut, or a population of microbes stays in your gut, is something on the order of 2 weeks. You kind of cycle around about that time scale. So that's a fairly stable relationship. So some examples of commensalism. So the K12 that's in your human intestinal tract, The bifidobacterium, it actually helps with your digestion. The E. coli in your intestinal tract is really not believed to be doing anything special for you. It's just there. And it's about 2% of your intestinal bacteria. So if you were to if you were to swab intestines and then spread it on a plate and put it in a normal oxygen atmosphere container, what you would get growing on that plate is E. coli. So it is the dominant facultative anaerobe in your gut, but it's not, by any stretch of the imagination, the major component of the gut bacteria. So parasitism, so plasmodium in blood, like malaria, that's one good example of that, where this can last many years, and it certainly does no benefit to the human being that has it. Pathogenesis is a slightly different situation, though, than parasitism, in the sense that pathogenesis is not a stable relationship. It's usually, highly detrimental to the host, but it's not something that sticks around for a long time. That's kind of what defines it as a distinction between it and parasitism, is just how long it lasts. So E. coli 0157H7, enterohemorrhagic E. coli, in your human intestinal tract, that's not something probably any of you have ever gotten. If you got it, it's real bad, like you're bleeding out your butt. It's bad. Okay, so this way of looking at things is a little bit problematic from the perspective of synthetic biology. And one very clear indication of why that is cows. So it turns out, if you were to do the same experiment of swabbing the intestines of a cow and spreading it on a plate in an oxygen atmosphere, you'd find a 157H7 inside their gut. And that is actually the reason why it gets into hamburger meat, is due to incorrect practices of how they prep the meat. So in the cow, this is not a pathogen. This is not a parasite. It's a commensal sort of relationship, just like this one. So in this case, there's nothing mechanistically distinct about something being in any of these particular categories. These are just descriptions. These are like clinical descriptions of the result. But the underlying genetics of what controls an interaction between a microbe and an animal, it's all the same stuff. So we're going to talk about what that stuff is and how it works. So just throw out these ideas. It's not really a reductionist view of biology. It's just a very descriptive, they're kind of made up abstractions that are based on phenomenological information, but at the molecular level, they have no predictive value. So in terms of the sort of things we can be talking about in the context of engineering therapeutics, or talking about what makes a pathogen, there are lots of different categories of things that you might be talking about. You have things like worms on one extreme, and on the other extreme you have prions, which, there's no DNA even involved in that. It's just a protein that does crazy stuff. So we can lump this into three categories based on just physicochemical composition. You have the prions all by themselves as being purely proteinaceous things. In this middle category, you have things that are non-cellular, meaning they don't have an aqueous cavity and they don't have replication, and they're dependent on a cell for replication. So you can have things like plasmids, transposons, viruses, and bacteriophages. All can potentially have these types of interactions with a host or play roles within these processes. Then at the other extreme, though, you have the cellular things, which could mean bacteria, could mean protozoa, so which are eukaryotic small single-celled organisms. And of course, you can get multicellular organisms, and you can even just be talking about mutant versions of normal cells that compete, and think of that as a pathogenic agent. So actually, interestingly, there's something dogs can get where it's a cancer of dog that has evolved to the point where it can cross from dog to dog. And they do this by having a heavily glycosylated surface. So actually, a cancer can become, in and of itself, a bona fide pathogen in the same sense that anything else is a pathogen, like it spreads organism to organism and bad news, results. Okay, so let's talk about this from a molecular perspective. If you have an interacting organism and you have some compartment, and we could be talking about an organ, a cell, a test tube, or a rock, doesn't matter what you're talking about, some other compartment, just in terms of just where things are relative to one another. Things can be not interacting at all, things can be interacting with the surface, or things can be inside. In that case, you might say it's barrier penetration. And if this is a closed compartment, how things get attached to the surface or inside the container is a meaningful question, because something has to happen to get around that barrier. It's not going to happen spontaneously. And the other constraint that you have is that you have to have growth. So you have to have a supply of all the atoms needed to replicate. You need to have an energy source, which probably is coming from the same thing as the atoms that you need to grow. There are various ions that are required, other trace elements such as iron. And if you were to look at any biological system, all these things are present. I mean, if you pick even what you would consider to be the lowest energy, most worthless areas of your body, like urine, you can burn urea. There is energy to be had in that molecule. So, there's not all this other stuff. You don't have much calcium, potassium, and it turns out iron is a significant thing in this space. These things are not present, so things don't tend to grow in urine. However, you look at any body fluid, they're going to be organic compounds, and those are all things that microbes can conceivably use for energy. So the big question, first of all, in all this space is, are you going to have a full set of all these various things present in that environment that would enable the microbe to survive? So in this sort of context, if you've got a microbe, say, inside a cell or inside an organ, in the bloodstream, wherever, conceivably everything is present for that microbe to survive. So it's like physicochemically possible to have survival in that environment. If you're outside of this environment, if you're on the extracellular region of this compartment, then obviously you can't survive because there's nothing out there, just a bunch of white space. So, you start looking at more fine-grained structure of what's happening with a cell. You might have organelles. Organelles don't have to have the same chemical composition as the rest of the cytoplasm. So there can be scenarios where an organelle might support the growth of a pathogen better than another compartment, and vice versa, where things couldn't survive in those particular compartments. And similarly, there is higher order structure if you're talking about an animal or plant system. You have multiple layers of barriers in the human body, right? And the next kind of level up is the level of organs in a human being. And that, again, doesn't have to be chemically related to what's inside the cell. You might have tons of glucose out here, but glucose itself is not usually something... it's like glucose phosphate, not glucose, inside the cell. It's chemically distinct. So, again, different environment, and different microbes might like that environment or might not like that environment. But suffice it to say, most organs are going to have most of the nutrients in there, but they might not be all in the same form. There might be some missing nutrients. There can be sequestration of particular types of nutrients to help avoid bacteria and such from growing in that environment. But right here there is a cell, and any way that microbe can get to the nutrients in that cell, there is the opportunity to have pathogenesis. So it really matters where the bacterium is, and really it's only the cytoplasm where everything must be present in some fashion. Like, you don't need to have iron necessarily inside your organs, but you absolutely need iron inside the cytoplasm. So the most kind of privileged environment we can talk about is going to be the cytoplasm of a cell. Okay, so that's the basic mechanics of all this, right? I mean, in a chemical sense, that's all that's going on with these relationships. Is you got a bacterium or a fungus or a parasite, whatever, that's trying to get at the nutrients that the cells contain. All right. But then things get a lot more complicated because this is a battle, right? I mean, you got all these nutrients, they're microbes trying to get at those nutrients, and if nothing else happened, we'd all be dead, right? They would eat all our nutrients, so we would just be bacteria. So what is kind of counterbalancing all this through evolution is lots of fancy mechanisms. The first one being iron sequestration. So iron is used by cells for multiple things. You know, you have iron sulfur clusters in your proteins. All these enzymes, like P450 enzymes, require an iron center to do the chemistry. So it is a fundamental building block. It cannot be substituted for some other atom. You need iron to survive. Every cell needs it. So what pretty much every biological system does is it sequesters the iron. You don't have free iron floating around. And part of this is to keep it away from bacteria. The other reason to do it, though, is iron's really reactive stuff in the context of all these organic molecules that are kind of high energy molecules. So chemistry would be happening spontaneously if you didn't sequester the iron. So that is presumably the other reason why this takes place. And the way it's sequestered is through binding it to proteins. So if you're talking about the blood system, there's a protein called transferrin. And transferrin is a free-floating protein in your bloodstream that specifically binds to iron. If you're talking about milk, there's iron in milk, plenty of it. It's a good nutrient for the baby. So it's in there, but to avoid microbial infection, what's in there is lactoferrin, which is a very similar protein that does the same thing. So you also have in your bloodstream heme, and that is an iron center associated with a small molecule. So that's another solution systems have, is to tie up the iron in the form of a small molecule complex. Okay. If you're talking about extracellular compartments, intracellularly you get different sorts of carrier proteins for the iron than you get in extracellular environments. It tends to be more like the iron complexes when you're talking about cytoplasm. When you're talking about extracellular environments, it tends to be these carrier proteins. Okay. Now, if you were to look at the genome of any particular bacterium, you're going to find solutions to dealing with this problem on the bacterial end. So the human being evolves the ability, actually humans didn't evolve this because horseshoe crabs even do. I mean, everything does this. This evolved billions of years ago, right? So every cell has got this defense against microbes taking over, but bacteria have responded to that and evolved processes for getting around it. And you call this high-affinity iron transport. And the details of what specifically is in the microbe matter here, because there are lots of different things that can be tying up iron inside the cell, right? If we're talking about milk, we're talking about a fundamentally different chemical composition of a lactoferrin iron complex than what exists if you're talking about the bloodstream. So you need kind of tailored solutions to working around this conundrum if you're a bacterium. So there are many different mechanisms of high-affinity iron transport. So if you look at what's in E. coli, there are actually two processes going on by which the bacterium is able to pick up iron. First of all, there is low-affinity iron transport. Low affinity meaning the concentration of iron that is required to have a significant, measurable flux through this pathway is on the order of 1 micromolar, which is a lot. And there are 3 transporters that can specifically transport free iron directly from the extracellular environment into the cytoplasm. Zoopty, mint, H, and FeO-ABC. So whenever the bacterium is in an environment that has these free ions, it just pumps them in. However, when it's being a commensal or a pathogen or whatever, it needs to do something else because there is no free iron in that environment. It's all tied up. So that requires the separate set of processes called high-affinity iron transport, of which there are several systems. So we're looking here at a diagram describing what's inside a pathogenic E. coli, not the lab strain of E. coli, but some of this is in the lab strain of E. coli. So the major things are enterobactin and citrate, and I'm not sure if these other guys are present in E. coli. Yeah, they are. The Fue is in E. coli. Enterobactin is not in K12, but it's in other E. colis and it's in many salmonellas. So enterobactin, though, is the main one. So this is a little chemical ligand. And I'm going to go through just what these chemicals look like so you can see what they're doing. But in general, what happens is the bacterium will either try to import in iron complexes. So when you have things like heme, where it's just iron attached to a small molecule, the bacterium will typically just transport in that entire complex and then degrade it in the cytoplasm. In the other cases, though, where you have an iron associated with a protein complex, a bacterium will secrete something called a siderophore, which basically means another small molecule that likes iron even better than the carrier protein. And those will spontaneously react in the extracellular environment, getting the iron into the siderophore, and then the bacterium specifically transports that small molecule iron complex. So that's what's going on here. Enterobactin is the most common siderophore that the E. coli's make, and it simply secretes this thing out, and then the receptor is specific for the iron complex of the siderophore. It pumps it in. You will notice that in all cases here, there are certain proteins that are always at play. So the EXB genes, and the big one is TUN-B. So actually, it's a very common means of eliminating pathogenic behavior in a microbe to just knock out TUN-B. So a lot of live vaccines are done that way. It's also a safety measure done in labs that study bacterial pathogenesis to just knock out TUN-B. And as long as you're studying in vitro behavior and not in vivo behavior, where you would need this to have in vivo behavior, you can largely eliminate the ability of the microbe to grow. However, how would you grow it? How do you grow the microbe if you knocked out this iron transport? Doesn't that seem like a problem? Huh? Okay, and why does that work? Okay. So as long as we don't muck with the low affinity iron transport, we can just keep feeding them free iron and grow them in LB media. And actually, there's plenty of free iron in LB media. That stuff has everything in it. There's nothing wrong with a TUN-B mutant of E. coli. They're perfectly happy, but they obviously have problems if you try to stick it inside an animal because no growth can happen. All right, so heme, that is going to be a major player in the story because you've got tons of iron tied up in heme in your bloodstream, and that is a very popular thing for microbes to try to get at. So in the case of heme, you're just going to transport that chemical directly through one of these receptors and then degrade it in the cytoplasm. These sort of siderophores look like this. So actually, these are polyketides. Actually, we've seen it before. When we were talking about non-ribosomal peptides and polyketides, there was a paper by David Liu where they had pulled one of the domains from the enterobactin biosynthetic pathway, and they were doing selection for restoring high-affinity iron transport. So you might look back at that paper to remind yourself of the relationship between these two stories. Enterobactin is considered, though, like the Rolls-Royce of the high-affinity iron transport world, in the sense that enterobactin has really high affinity. This is something, I believe, 10 to the minus 15th molar affinity. If you're not transferrin, I think it's, I want to say 10 to the minus 12th affinity. So this can pull iron out of transferrin. Enterobactin is something like 10 to the minus 20th affinity. It's like really good. So when you look at really aggressive pathogens of bacteria, they will typically have enterobactin, and it's a very small minority of the E. coli's that have that pathway. So it's, you know, if you wanted to kind of rank these virulence factor genes, these things associated with these secondary processes of cell-cell interactions, this would be kind of in the high category in terms of dangerousness to be working with. So in terms of how this stuff is biosynthesized, it's a polyketide kind of process. So you just build it up. It's just normal chemistry. It's branching off charismate. A bunch of enzymes happen. They get you to this monomer unit. And then that monomer unit gets polymerized into the final siderophore. But there are lots of variations on siderophores. Different microbes make different structures. They only really have to have the general property that they like to bind iron, and binding iron really is just tantamount to having some Lewis bases in the right position, so things that can donate electrons. Okay, so the story gets more complicated though. So if you're talking about normal strains of E. coli, if I were to squirt that into your bloodstream, well, lots of bad things could happen, but actually it's not going to grow in your bloodstream, even though it has enterobactin. And the reason for that is there are proteins such as this, lipocalin. Lipocalin specifically binds enterobactin. This is something that your body, at some point, and actually I think all mammals have this. I think it's somewhere around like the mammal breaking off from the rest of the family tree that this mechanism has evolved. So this is specifically your body having a dedicated protein to deal with blocking E. coli from growing in your bloodstream. And if you think about that, that makes a lot of sense that your body would have a lot of E. coli specific defenses because this is the dominant bacterium that is in your gut, and you eat something sharp, you're going to get cut, you're going to get E. coli into your bloodstream, and obviously that is going to happen spontaneously often in your lifetime. So you have to have some way of dealing with the fact that the E. coli has these genes in it. So this thing ties it up, and therefore there's nothing to transport back into the microbes, so there's nothing to eat. So if you look at some other kind of enterobacterial species, like many of the salmonellas, they will do additional things to their normal enterobactin to prevent this. So one very simple thing that you'll see is just adding a sugar moiety. You add a sugar moiety, it's a different structure. If it blocks binding to these lipocalin things, then it's back in business and it'll work. It can pull iron and transport. So again, these are genes that are in that really high category of dangerousness because there isn't a defense against these chemical structures in the body. Okay. So the second major defense that biological systems use against microbes is physical barriers. So minimally, you have the lipid bilayer that is preventing things from directly getting at the cytoplasm. So that's a barrier that you have to get past somehow. Many also have a wall. If you're talking about a gram-positive bacteria, you have this S layer of carbohydrates all over the surface. If you're talking about things like plant cells, you have a bona fide wall made of cellulose that's very hard to get through. And then, of course, you have means of getting past these barriers in some microbes. So if you have a multicellular organism, you can have additional barriers that are like specifically encoded. So if we're talking about a cell that is a chemical barrier on the surface, a carbohydrate layer typically, if you're talking about one scale up in the organization of an animal or plant, you are talking about cellular or extracellular matrix-derived barriers. So like your skin, that's dead cells, right? That's not just chemicals, they're actually cellular structures associated with that barrier. Okay. So your skin is an example of a specialized barrier in the sense that the whole purpose for it kind of existing is to put this wrapper around your body that prevents things from getting in. And nothing really can get through skin except through physically breaking it. It's a very, very effective barrier to pretty much everything except something like a worm that can burrow its way through your skin. So if you're talking about bacterial infections, I'm not aware of a single bacterium that it can actually get through your skin. They're always opportunistic infections in the sense that they either have to get through a cut in your skin or you have to ingest them or breathe them in and get them through some epithelial route. So there's a major issue that microbes have to overcome, that they just can't get through your skin. Okay. So from the perspective of a bacterium, this is what the human body looks like. You're a big sack of delicious nutrients with two access points. You have your mouth, nose, ears, and eye socket that are entry points that things are constantly being put into. And you have your anus, urethra, or vagina where things are coming out. And those are the places where most infections take place, simply because every other part of your body is surrounded by skin. And the only way to get through the skin is physically breaking the skin. So, the thing that protects most of the organs of your body is an additional barrier, that is the epithelial layers that surround all your tissues. So you have multiple levels of barriers that all must be crossed to have a successful pathogenic infection. So you got your skin out there, So inside this space, though, that is actually accessible to the extracellular environment and just protected by epithelial layers, you have your stomach, your lungs, and intestines. And those things are chock full of bacteria. And the reason for that is they are directly exposed to the environment. When you eat something, there's going to be bacteria all over the food, so that's going to get into your intestines, and from your your stomach, and from your stomach, it goes to the intestines. Now, in the stomach, you got this really low pH barrier. So a lot of stuff gets killed off, but of course you can have things like probiotics because some things can survive in the stomach. There are additional defense mechanisms in the bacteria to deal with the fact that it's a very low pH environment. But the default would be things would just die going through there. Then you have some things that are just a little bit harder to get to. So things like your kidneys, those actually are exposed to the extracellular environment, and you can get microbes that are capable of going through your ureal tract and up into the kidneys, and that's one of the really bad E. coli's. It will do that, EPEC, Pathogenic E. coli. So yeah, those are actually accessible environments. They're not blocked off from the world. But then you got other things that truly are blocked off, like there is no entry point that doesn't involve passing through an epithelial layer. So your blood, your lymph, your liver, your brain, these are all much harder to get to. However, every single one of these environments can get infected through some microbe. And finally, inside these organs, you have your cells, which of course have their own cellular membrane. So it's many layers of barriers to get to most of these juicy nutrient sources. So where do you find bacteria? You find them all over your skin. You find them all over this whole middle region of your body that is exposed to the environment. And occasionally you'll get bacteria in things like the kidneys and the bladder. And in general, that is where, that's bad news when bacteria get into that environment. And those are very common sorts of infections for a human being to get. But your body is very actively trying to clear bacteria from those environments. If you were to look in places like the blood, the liver, the brain, there are no bacteria in those places. Those are actually sterile environments. It's hard for the bacteria to get in, and they're very aggressive immune responses to getting the bacteria out. So it's actually very, very difficult to get a microbe to survive in those places. Okay. So how do you get past these barriers? Eating and drinking, that'll do it. At least for the ones that are just like your gastrointestinal tract. But the main issue is getting past the skin, and you can't get past the skin unless you're something like a worm with teeth that can burrow through it. I'm not aware of a bacterium that can do it. Potentially you could get something that was like irritation. I don't know. I'm not aware of a case of this happening. So here's how a lot of things get in. So the sort of things that in the modern world, in the Western world at least, that you actually get infected with bacteria-wise are things like the pathogenic bacteria, Shigella, Salmonella, E. coli, pathogenic E. coli's. And all those things typically get in through the same route. So you eat something, the bacteria is on it, and in your intestine, so it survives through the stomach because it has acid resistance genes, gets into your intestine, where this is kind of a model of the epithelial layer in your intestines. And you get these specialized cells called M cells, which actually are part of your immune system in the sense that these things are meant to pull in bacteria, kind of sample the epitopes present on the bacteria so that your body can train itself to recognize those things. However, this is also a point where bacteria can infiltrate. So if you're talking about a Shigella infection, what happens is the normal route going through here, but then the bacterium pops itself out of a macrophage that should be intercepting it, and then it can zip around in your bloodstream, it can zip around in the epithelial layer, it can actually cross epithelial boundaries by doing this thing with an actin. It polymerizes actin on one pole of the rod, and it can kind of shoot around like a bullet and just pop through barriers by doing that. So if you look at this initial process of invasion, where a bacterium is able to invade M cells and go through it, the typical solution to how bacteria do that is through using type 3 secretion apparatuses, which I believe we've talked about before, or at least we talked about flagella as being this big protein complex. We did talk about type 3 secretion in the context of protein secretion mechanisms. So this is many genes, a big complex series of regulation to guarantee that this assembles properly and is only expressed when the bacteria really needs it. So you got all that stuff going on inside this thing, and all this regulation that is helping the bacterium know that it's actually in this environment where it needs to use this. So the fourth defense is phagocytosis, where you have specialized cells like macrophages, neutrophils, that are going to specifically eat debris inside your body. So these things are responsible for keeping everything clean. If you were to squirt some dust into your bloodstream, actually that's a bad example because those are cells. If you were to squirt like talcum powder into your bloodstream, it's the macrophages that would clean that up. There's a huge population of them in your liver called the Kupffer macrophages that deal with most of the bacterial infections. So if you squirt bacteria in the bloodstream, most of it ends up inside these cells in the liver. Okay. So there are mechanisms of evading this too. So, and we'll get to that evasion mechanism because in a second, but there's a second piece of this that is the complement system. So the complement system is kind of like the thrombin system in your bloodstream in the sense that it's just made of protein. There's no genes involved. I mean, obviously some genes in some organ somewhere are biosynthesizing these proteins and secreting them, but the system itself is not dynamic with respect to transcription. It is a purely post-translational system where when you get some chemical entity with a particular chemical pattern, and there are many different patterns that this system recognizes, but it tends to be glycotopes, so the sugars that seem to be on the outer membrane lipids of various bacteria. Things called lectins, so things that bind those things will first settle down on the surface, and then you'll have this cascade of interactions of deposition of these complement proteins that ultimately results in the formation of a pore that destroys a thing. So there's some fancy biochemistry going on here, but this thing is ancient. Like horseshoe crabs, which have been around since like billions of years ago, they have complement in them even. So this is not a human-specific thing. The things with the macrophages and such, that not all things have that, but like every animal has a complement system. The sixth defense, though, is adaptive immunity, where not only does your body have this intrinsic capability to recognize specific pathogenic patterns, your body can evolve antibodies and T cells that specifically recognize new epitopes and have dedicated pathways to destroying specific things. So how do bacteria deal with all this? Because obviously you can still get a bacterial infection, And the main way they do it is things called capsular polysaccharides. We've seen these before. We talked about O antigens in the context of mechanisms for producing polysaccharides. This is what they make these things for, is to decorate the surface with a pattern of sugars that makes it look like something that belongs in a human body. So this is a micrograph of a pathogenic E. coli, and you'll see several layers. The membrane actually is right here. So you have two additional layers of carbohydrates. In this case, it's a K capsule here, which is usually polysialic acid, if you're talking about things that infect humans. But there are many different categories of these things. And then this guy has something called a non-fibrillar adhesive, and actually this is the only paper I've even seen describing this virulence factor. But it's presumably something like a pilus in the sense that it enables the bacteria to stick to some other cell type, but it looks under a micrograph like a big fuzzy shell that's extending beyond the K capsule. So who knows what that is? But there are other mechanisms that bacteria use. Like there's one bacteria that puts a protein on its surface that reacts with the albumin in your blood, coats itself in albumin, and therefore it just looks like a very small blood clot, and that helps it evade immune responses. There are also lots of genes out there for doing weird things to antibodies. So sometimes they'll catalytically cleave them, sometimes they'll just bind them. So if you bind an antibody to your surface, but you bind it in the wrong conformation, like you bind to the VC domains, like the constant domains, then just sterically, the other part of it is not available to bind, and you don't get the cascade of events that you would get if you had it binding through its correct orientation. So there are lots of additional tricks that bacteria use in concert with these processes. So you'll see quorum sensing systems in a lot of pathogenic bacteria. So why exactly that is often unclear. To some degree, it's just sensing what environment are they in so they can decide, is it time to make a type 3 secretion apparatus, or should I just eat while I'm here? Questions like that. But certainly people have claimed that this could be part of coordinated attacks. You can get things like biofilm formation, so that's very well known in Pseudomonas aeruginosa, where that will, you'll get this biofilm in your lungs that is much more resistant to antibiotic treatment than if there was no biofilm. So there's kind of a community effects that you can get through cell-cell communication through all this, and people are still just working out just what even exists within this space of additional mechanisms that the microbes are using to survive in these places. You'll get lots of things that like to stay inside macrophages. So for example, Salmonella starts its infection cycle the same way as Shigella by invading an M cell. It goes ahead and gets inside the macrophage just like any other bacteria is supposed to do. But the result of all of it is a second type 3 secretion apparatus turns on inside the macrophage. The bacterium starts secreting proteins into the cytoplasm of the macrophage that change the composition of the vacuole so that it no longer will fuse with liposomes. So it's a stable situation for the bacteria, so they can just float around inside a macrophage and go all over your body. And then when they find some new place that seems desirable to them, they have the ability to burst back out of that vacuole and create an infection somewhere else in your body. So bloodborne salmonella is not a good thing. Yeah. There can be resistance to acidity in the stomach that seems to be required for a successful eating-based mechanism of infection. Secreting outer membrane vesicles. These are mechanisms that have been observed and they've been studied. What exactly they are for, who knows? There are all sorts of mechanisms of adherence. So if you have type 1 pili, that is what all these things are using to stick to the walls of your epithelia in your gut. If you have P. pili, which we saw in an earlier lecture associated with phase switching, those things bind to a glycotope in your kidneys. So that's for pyelonephritis, you need that. If you're talking about the enterohemorrhagic E. coli, the one that's in the hamburgers, that does this really cool mechanism where it's 40 kb of genes just to do this, but the bacterial will first express a pili that allows them to adhere to the epithelial cells in your gut. but then you have a type 3 secretion system that secretes effectors through both membranes into the epithelial cell that put kind of adapter proteins on the other side that the bacteria kind of joins up another protein to. So you kind of lock the bacteria to the surface of the epithelial cell, and then it secretes effectors that cause actin polymerization that make this pedestal happen underneath it. So it ends up kind of sticking out, bathing in the nutrients of your body. So all sorts of fancy things out there and all sorts of sources for genes. So one of the big benefits of all this stuff, you got all this fancy evolution happening, you got a battle where the bacteria invents some new thing and then the body evolves some mechanism for fighting that thing, so it's, it's an arms race. And the result of this is lots of chemical diversity, lots of mechanistic diversity has emerged from this, so it's kind of a treasure trove of genes for doing synthetic biology. And a lot of this stuff is just useful transcription factors and useful sensors, but also the sorts of genes that should enable us eventually to build things like targeted probiotics and therapeutic organisms. All right, any questions? Alrighty, see you all Wednesday.